Welcome to this session organized by Aalto University, Finland, University of Chieti Pescara in Italy, and University of Tübingen in Germany. We will introduce the ConnectToBrain project. In the slide you see the full name, Connecting to the Networks of the Human Brain. The project, the ConnectToBrain project, started last fall with European Union funding. European Research Council, or the ERC, is the funding agency. It is a so-called synergy project with 10 million euro funding for 6 years. That is until 2025. Our plan is to build technology and artificial intelligence that would enable a paradigm shift in neuromodulation therapy using TMS, or transcranial magnetic stimulation. The device technology will include a multi-locus stimulator, meaning that we can stimulate many different places at short time intervals. And so we can target the pulse, the magnetic stimulation pulse, to different brain regions electronically without moving the coil system. And this change of target can be done at very short time intervals, shorter than 1 millisecond. To control the targeting and timing of the pulses, we will develop the use of concurrent EEG that will monitor the brain state, brain activity, to provide real-time feedback from the brain. To reach the goal of the project, we will need to develop new signal processing and artificial intelligence methods. But before going to that, perhaps Ulf Ziemann could explain what the project's final goal is. So please, Ulf.
Yeah, thank you, Risto, for this introduction. We have already demonstrated that EEG-defined brain states, such as the phase of the ongoing oscillations in the brain, for example, the sensory motor mu rhythm, determine success of induction of long-term plasticity in the human cortex by repetitive TMS. So for example, LTP, long-term potentiation-like corticospinal plasticity with a 100 Hertz triplet burst stimulation is induced only if the bursts are synchronized with the negative phase of a mu rhythm, a state of high corticospinal excitability, as has been indexed by large motor evoked potential amplitudes. In contrast, the identical stimulation synchronized with the positive phase of a mu rhythm, a state of low excitability, or random phase stimulation does not result in corticospinal plasticity. These findings tell us that the instantaneous brain state at the time of the TMS pulses is critical for magnitude and direction of long-term synaptic change. We have now started in the clinical arena to treat patients after motor stroke and also patients with major depression disorder using this new technology. In ConnectToBrain, the ultimate goal is to develop closed-loop stimulation of complex distributed networks, such as the frontoparietal working memory network in Alzheimer's disease, or the distributed motor network in stroke patients with arm-hand paresis. Closed-loop approaches, almost done. Closed-loop approaches mean that we measure and analyze the effects of brain stimulation online to predict the effect of the next pulse and optimize the stimulation protocol iteratively to achieve the best therapeutic success. That means restoration of a dysfunctional network. To be able to perform closed-loop stimulation, however, in a way that optimally modulates the brain, we need very fast real-time signal analysis and artificial intelligence algorithms that decide how the stimulation should proceed. Gianluca Romani, how do you see the prospects in succeeding in this very demanding endeavor?
Thanks, Ulf. Well, actually, signal processing in the brain is very fast. The time scale spans from milliseconds to tens of milliseconds. So the challenges are to pre-process and analyze brain signals very fast. And we will do that using data recorded by high-density EEG to identify brain connectivity states in real time and to determine how the stimulation sequence should continue. Brain activity and brain connectivity will be derived in real time using efficient strategies, able to predict the evolution of brain states with milliseconds resolution. To help this estimation, we will use as priors information from different neuroimaging modalities, such as MEG or function MRI, applied to the same subjects, or from available neuroimaging databases. The key technology will be the development of devoted software algorithms to control the stimulation sequences. The typical algorithm then will start at preselected sites of the brain, and depending on observed effects on brain connectivity, will automatically move the stimulation target in order to approach brain modulation or therapeutic goals. The approach, as said, will be based on artificial intelligence strategies, such as a machine learning framework. This will permit to automatically derive relevant features in the brain state's changes induced by the stimulation, will transform these features into new driving inputs for the simulator module. Risto, Mr. Moniemi, would you like to introduce the next 3 talks in view of the general structures of the C2B project?
You're on mute.
Risto, you have to unmute your microphone.
Okay, now you should be able to listen. Sorry for that. Thanks, Ulf and Gianluca, for nice introductions. Before I go forward, I would like to just mention my enthusiasm about this possibility of connecting to the human brain magnetically. So the stimulator system, the artificial intelligence system, and the brain will be just one system that is interconnected. And the big challenge, as Gianluca mentioned, is creating the algorithms to control this. And the purpose is to be able to do something that will provide a new kind of and more effective therapy that we have at the moment, as Ulf emphasized. So next we're going to do some of the details of the project. First, Timo Roine and Jaakko Nieminen will explain the basic technology. After that, Laura Marzetti from Chieti will explain the signal processing artificial intelligence part that we are about to develop. And finally, Christoph Zrenner from the University of Tübingen will talk about the neuroscience developments. So we will start with Timo Roine, and you have the microphone off, I hope.
Yes, thank you. So me and Jaakko, we will talk about the development of the multi-locus closed-loop TMS system. And I will start with some very basic details about TMS, transcranial magnetic stimulation. Is a method where we can activate the brain non-invasively and painlessly through electromagnetic induction. And it is currently used in the clinic for testing the integrity of motor pathways and also for motor language mapping, for example, before surgery, and for treatment of several neurological and psychiatric disorders. However, the present technology is limited because we can only target a single site at the same time, and it's also very operator dependent, and the target is very slow to change. and it's also blind to ongoing brain activity. These all lead to rather low therapeutic efficacy at the moment. I will show you a video. This is an artist's illustration about the final product, which would be realized in the end of the project. Okay, so next I propose that Jaakko Nieminen will present the development of the multilocus coil technology.
Very good, thank you. So I'm Jaakko Nieminen from Aalto University, and I'll introduce you the transducers development that we're conducting in this project. So it's about coils and coil design. So our aim is to achieve electronic control of the stimulated cortical site. So and that would have the benefit that we would be able to adjust very fast to the stimulated cortical site without any mechanical or manual coil movement. And to achieve this, we use a set of overlapping coils that in which we adjust the relative current strengths in order to change the induced electric field pattern in the cortex electronically. We design these coils with an optimization algorithm in which we first start by defining the overall coil shape. So we may start with a planar coil, for instance. Here's a planar surface. We form a triangular mesh of the surface. So discretize this surface. It could be any other shape as well. And we look for the surface current density on this surface that most efficiently activates a given target in the cortex. And to solve this, to find out what is the winding pattern that we need to achieve that stimulation, we look for the currents that circulate around each node of the mesh. And finally, we from the contours of the surface current density, we obtain the final coil windings. And this same principle that we first applied to designing single coils for conventional TMS can be then converted to a design framework for multi-locus TMS, in which we design a set of coils. For instance, here is a set of 5 coils that are stacked on top of each other, each shown schematically on these figures here on the top. So here red means clockwise and dash blue means counterclockwise circulating currents. So there are, in this stack, we, for instance, the algorithm gave us as figure of 8-like coils, 2 that are in perpendicular orientations, a round coil, and 2 other more complicated coil patterns. And now if you look at that, what kind of electric field patterns these coils would induce in the cortex, we would obtain electric field patterns like this. So figure of 8 coil would induce the electric field maximum underneath the coil center. The other one would induce the maximum underneath the same location, at the same location, but in rotated direction. The circular coil would produce a donut-like stimulation pattern, and the other 2 coils would produce other field patterns. Well, as such, these field patterns in the cortex are not really that useful when you think. But they become highly useful when we start combining these with each other. So when we apply current to several of these coils, at the same time as illustrated here on the bottom row, where we can then electronically translate and rotate the peak-induced electric field in the cortex. So here, for instance, in this case, we are able to move the electric field maximum within a roughly 30 millimeter diameter cortical region in a spherical head model. So given this design, we proceeded and constructed this coil set as illustrated here, and finally measured the electric field pattern induced by each of these coils. And these patterns quite well resemble the ones that we thought they would be doing. With 5 coils, we can do already quite a few things. One example that is useful in practice is compensation of head movement. So typically with the conventional coil, one would need to, when a patient or subject moves their head, one would need to follow the head movement by manually moving the coil in order to keep the stimulation to the site intact. But with the multi-locus transducer, one could then electronically compensate for that movement, and think that if the head moves, one can adjust the currents in real time such that the stimulated cortical site would remain intact. Another benefit of multi-locus transducer is that one may automate different procedures. For instance, automate the search of stimulation targets. On this slide, I show you an example where we placed a 2-coil transducer here on top of the motor cortex, such that we could electronically translate the peak-induced electric field on the motor cortex, and we used this to find the stimulation target that best activates a given hand muscle, leading to maximal MEP or motor evoked potential amplitudes. For this, we developed Bayesian optimization method in which we sample or stimulate different locations of the motor cortex in an adaptive fashion, taking into account the information that and the responses that we have gathered during the previous stimuli, in order to minimize the number of stimuli or the number of time needed to find out the location that produces the largest muscle response. And this work has been funded by several sources. Now Timo will continue from this.
Okay. So in the ConnectToBrain project, we will the ultimate goal is to develop new MTMS therapies, but to reach these, we have these 3 stages, or the three different groups working on the problem. So these groups are related in many ways, but one way is to think about it from the neurophysiological side. So first we think how we would how should the network be changed to be able to help the patient, and then we develop methods to actually modulate the brain into that target state, and then we try to build a system capable of doing this. But of course, also the relationship goes the other way. So what is possible to do with the hardware and software is also affecting the result of the two other groups. The whole system would look something like this. So we have the MTMS system here on the right top corner, and we stimulate the brain and measure EEG data, and then we analyze the EEG data with, for example, inverse problems and connectivity metrics based on the EEG data, but also using prior data from MEG and fMRI and diffusion MRI. Then we extract features or use machine learning and AI to get the next stimulus parameters, and this forms a closed loop working to control the brain towards a desired brain state. In the hardware and software group, in addition to the development of the transducers or the coils, we are working on several other tasks. One of which is very essential is the electronics to control the transducers, but also the neuronavigation to accurately be able to position the new coil also with respect to the head, and also new EEG technologies is another part of the project. Robotics may be needed at least to position some coil versions with respect to the head, and of course the software needs to be able to do and integrate all of these separate systems and take into account also the methods and algorithms from the other groups. In addition, we have other related projects regarding current density imaging and MTMS fMRI, so doing TMS inside the MRI scanner. Regarding the electronics design, we are Heikki Sinisalo is working on this part of the project, and the idea is to use flexible and modular designs, so we can easily expand the system. Because of the high voltages, safety is a very high priority, of course, and also because of the many channels of the final instrument, also the physical size may be a constraint, so we have to aim to use compact modules. Regarding the software architecture, we are currently implementing the first versions of the software, and we are using this kind of publisher-subscriber model implemented with Kafka. Of course, the system has real-time constraints, so we should be able to do all data transmission and also all the algorithm processing very quickly, because the brain state evolves so quickly. Also, the system is based on very high modularity, so we can easily replace any element from the software with another. For example, at the first stage, we may use simulated EEG data or pre-recorded EEG data to test the system, and we can easily replace the EEG system with a simulator component. We are also doing user experience research. This is a video about tractography informed neuronavigation. So Baran Aydogan and Victor Souza are working on this part of the project. And this is a method which can in real time measure the connectivity of the point stimulation locus and visualize it in real time also. So here this work was presented in Berlin last year. Then about TMS and fMRI, we have Mikko Nyrhinen working on this part of the project. He's working both with the TMS fMRI coil, but also with the impulse noise measurements inside the MRI scanner. And in addition to that, we are working on MTMS and fMRI in rats. This is the work of Juuso Korhonen and Victor Souza, presented also last year. And this is the whole hardware software team. Actually, this photo shows most of the ConnectToBrain project, but the names listed here are working in the hardware software team. And next, Laura Marsetti will present her part of the project.
Thanks, Timo. Actually, we have 4 minutes for questions. If there
really?
are any from the audience, you can either use the chat or you can just turn the mic on and ask. There's one question posed in the chat I already answered. Many people ask whether the different coils in a multi-coil set interact and make it harder to control. Well, in this 5-coil system, all the coils are mutually, the mutual inductance is 0, so their electrical or magnetic interaction is very small. In larger system, this may be an issue, but I don't think that's a problem, a big problem.
Okay. If there's nothing else, then it's Laura Marsetti's turn.
So thanks, Timo. Okay, so my task this afternoon, at least in Italy, is to talk to you about what we are developing in terms of the closed loop part of the system that Timo has shown. And specifically, like the major aim of the closed loop system is that it will require, in the end, what we will design, the ability to modulate the brain dynamics toward the target state, which will be desired due to clinical, for instance, needs and so on. So the idea is to achieve this by a feedback-controlled approach, which requires as fundamental steps those of estimating EEG connectivity. So it's not anatomical connectivity as we were seeing before, but connectivity measured in terms of functional connectivity from EEG. But at the same time, what we want to achieve is robust methods to extract brain states from this information about EEG activity and connectivity, and being also able at the same time to design models on which we can rely for prediction on what the brain reaction will be after being targeted by TMS. So in order to achieve this, as I said, one of the first steps is to understand how to use the currently available methods for tracking dynamical connectivity from EEG and to make them work in the way that we need for the real-time brain stimulation. Usually, this dynamic functional connectivity estimation from any neurophysiological data relies on a sliding window approach. And this brings with itself a kind of a compromise, because on the one hand, we want short window length in order to be able to fast track the stimulation induced, for instance, connectivity changes in the project or also to different aims. But on the other hand, the shorter the window, the lower the accuracy with which we will be able to estimate our functional connectivity. So our first question, in order to understand how to deal with the amount of data that the project will deliver us, is which is the minimum window length of data that we will need in order to estimate functional connectivity. To answer these questions, we relied on simulations, which are really biologically inspired and designed in such a way to mimic brain activity in a realistic setting. And And so we divided our simulated data into, let's say, segments of length L, and assessed through the most common approaches, which is the length of L that we need in order to have a reliable estimation. The most two common families of methods for estimating functional connectivity are a class which is Fourier-based and a class which is Hilbert transform-based. So I will start with the Fourier-based approach, which, as you can see from this slide, do not only require us to define a length of the window of data that we want to analyze for estimating functional connectivity, but they do also require to chunk our L-long data into smaller pieces of length W. And in order then to average across this W and obtain the a Fourier-based estimation of our functional connectivity. So all methods based on Fourier, and here on the top you have a list of possible metrics currently used in EEG and MEG framework for deriving functional connectivity. All of these methods, as I said, need us to define a length of the window W, given also the data length, the full data length that we have available. So the data length here is depicted on the x-axis, and the window length is depicted on the y-axis. And choosing the window length also means that we are somehow choosing the frequency resolution at which we want to sample our functional connectivity. So for instance, if we take one of these methods, which is for instance the dotted red curve that I have here, I can read this plot as a chart, which tells me that, for instance, if I have data which contain 10 cycles of my oscillation, then I can obtain 80% of true positive rate if I use a window W of length 5 cycles. What does it mean to get 80% of true positive rate? It means that we are able to recover at least the 80% of the data that we simulated in our simulation. This, for instance, for an oscillation of 10 Hz would mean that we need actually 1 second of data to get a reliable estimation. This is, of course, a huge number, and for this reason, we also expected what can be achieved by using Hilbert-based estimation. I won't go too much into the detail, but the idea here is that I do not need anymore to define a window length W, and I will only observe what is the true positive rate as a function of the full data length. And here, if I concentrate myself on the same oscillation at 10 Hz, I can get an estimate of 80% of the true detected connectivities with only 3 cycles of data length, which is about at 10 Hz 300 milliseconds, meaning that it's less than 1/3 than I needed with Fourier-based approaches. So message number 1 of this presentation is that, okay, we have just started on this. We need to further inspect this work, but it's worth inspecting the Hilbert-based approach because we can bring that to a level which will give us the right sampling that we need in time. Then the second crucial point is, okay, so say that we have been able to derive these connectomes between all brain regions with an accurate temporal resolution, then we would still need to map this connectome into brain connectivity states, which are depicted here as these graphs on the bottom. And but that's not that easy because, of course, in simulation life is easy, but if you start having noise or if you start having data coming from different populations, then it might be quite challenging to assess to what brain state an observed connectome belongs to. So for this reason, we designed the framework in which we will rely on machine learning for doing this. And the idea is that if we have one connectome at a given time point, that can be thought as a point in an hyperspace. And in this hyperspace, what we want to do is to cluster all points in such a way that each of the clusters that we obtain, each of the sectors in this view that you have here, will be associated with a specific brain connectivity state, as in this case. And the state will be then representative of a family of connectomes. So how to perform this partition, as I said, through basically clustering algorithms and therefore through machine learning. But in order to understand which are the algorithms which might give us the best performance or good enough performance that we need in this framework, we again relied to begin with on simulations. And our simulations here start by defining a predefined sets and sequence of brain states, as you can see in the first row over here. So we are somehow designing which regions are connected and how these connections change over time in terms of brain states. And from that, we can map backwards our connectivity matrices and then apply on these connectivity matrices our clustering algorithms in order to test if the algorithms are able to recover the original brain state from which they were drawn. So as a first step, we need our algorithm to be able to do a very basic task, which should be to understand how many brain connectivity states were simulated in our case. And for this, we relied on a class of different metrics, which are the ones that you can find here on the x-axis, and you can see, like on the y-axis, the number of times in which basically the percentage of it, so the number of times in which they were able to detect the correct number of brain states. As you can see, I am showing different algorithms, but the performance do not much vary with the algorithm itself. Rather, we have clearly a family of methods which is worth further exploring, and some other which maybe it's not really useful to try to apply in our framework. Once we have defined the number of brain states as an input to our algorithms, we then want to further understand which among at least the tested algorithms will be able to give us what we want. And what we want is, on the one hand, to reconstruct the topology of the brain state, and on the other hand, to reconstruct the dynamics of the brain states. So we designed two indexes. One is a similarity index, and another is a percentage error in the dynamics. And we have, like on the x-axis, the lifetime of a brain state. And we are like showing basically the different performance of the different algorithms, which I won't go into the detail too much. But the idea here is that we will further explore these aspects in order to derive the algorithm with the best similarity and the lower error in dynamics, of course. This is really important as a step because this paves the way for the real-time estimation of brain activity part. So basically what we want to do here is to define the best set, and that can be done in simulation as we are doing now, but also based on real data and a priori information, as we were mentioning before, in order to derive which are the sets as a kind of basis function for our brain to like switch from one to the other. And also to understand after the intervention through TMS, which are the probabilities of our brain to change from one state to another, and so on and so forth. Another aspect that we are currently investigating, mainly by Riku, Lina, and Alto, is to really work on the more modeling part in order to being able to reproduce the TMS EEG dynamics. So these Kuramoto models are basically a computationally simulated ensemble of coupled oscillators, which for us are brain regions, and they provide you with a matrix which changes over time, which gives you the strength of the coupling between the different oscillations. So the first goal of this part of the work is to determine K, which captures the same temporal and spatial information of the TMS EEG data. So the first attempt was made on data in which left primary motor cortex was stimulated, and on the top row you can see the real data, the EEG data after TMS. TMS is 0 in this time axis, and two examples at 14 milliseconds and 47 milliseconds. while this is obtained through minimum norm estimates of the EEG data. While on the bottom row, you can see how the model was able, with the proper parameter, to reproduce these like topographies at the right instant in time. So the idea behind this is, again, that we could determine this coupling in the models for a number of relevant brain states, which are somehow the basis for, in the real-time setting, determine the dynamics of K to be used for predicting future brain states from what we know from the past and what we know from the current situation and from in the modeling framework, but also in the real data framework. So to wrap up, really the work of this part is now concentrated on building simulation, building models, and using offline data in order to understand which are the best approaches that we could use in order to design this basis function, which will be then used to allow the real-time framework to identify the brain states. And with this, I thank you, and I also show the methods team, which is the list of names shown in the slides, and a nice picture of the most of us, at least at the kickoff meeting. And I leave it to Christoph.
Thank you, Laura. Are there any questions for Laura before I start?
There's one question, at least
maybe
in the chat. Can you obtain artifact-free signal from EEG electrodes underneath the stimulation coil? And what is the material of the electrode? Who will answer?
I mean, what I've been showing is simulations, but for the real data, I think for what I know, I mean, I'm coming more from the methods in MEG as a personal history, but from what I know, if you discard like the first milliseconds after the stimulation, after the stimulation, then you are quite safe. So the question here is really when we need to use our data.
You need to have a proper kind of EEG, TMS compatible EEG. Then there may be artifacts from cranial muscles, and there are several other sources of artifacts. So you really have to do it in the right way. It's a very long story to explain. But in many cases, one can obtain high quality signals. On the other hand, artifacts have to be taken very seriously in TMS EEG studies. Christoph, do you have any further comments on this?
No, just, I mean, about the electrodes, typically the issue is having them flat enough so that you don't increase the distance to the TMS coil, and standard silver chloride sintered rings with gel work very well. Okay. Then it's my pleasure to talk about the neuroscience and clinical part, which is basically the question of what would we like to do if we have such a beautiful device. And I think it's kind of a dream situation to be in. You have a device that allows you to modulate brain networks in a very flexible way. And with that comes the challenge to define, okay, what do we actually want to do that will then help patients recover from diseases in the motor and memory systems. So we will work on these three stages. First with open loop data, where we can already do quite a lot of post hoc analysis to try and help the other groups as well with figuring out brain states that then inform the system. And then we will work with state triggered TMS. So this is where the brain state determines when we stimulate, but it's a simple state triggering. There is no closed loop interaction between the brain and the system where the stimulation changes over time to achieve a state, and that then happens in this later phase of the project. And for today, I will share with you 4 pieces of ongoing work that work with this open loop and real-time EEG triggered data. 2 that analyze actually a very simple data set, which is just an open loop TMS EEG simultaneous data set, which is, however, extremely useful, as I hope you'll agree when I share some of that data. And then moving from a single coil to 2 independent coils. And that's sort of an intermediate step to having this multi-locus TMS, having two separate coils where we can already do some science leading up to then MTMS, and of course the clinical applications with some of the proof of concept studies that have been done already or are ongoing. Okay, so starting with the first question, this is the question of what is the meaningful brain state, and this is work from Johanna Metzuma, who is currently in Tübingen. And it's something that Natalie Schaberonkov, who's now in Brad Wojtek's lab in San Diego, has started when she was in Tübingen. And this is sort of asking the question: if we know the state of the brain at a given time because of the evoked potentials. In this case, it's the evoked muscle potentials, and this TMS pulse probes the state at the time. If we know that, what can we decode from the preceding EEG activity? So I've drawn it on the right side. You can see the muscle potential. I'm highlighting it here. This is the evoked EMG when you stimulate in the left motor cortex, and the magnitude of this potential, it tells you, is the brain at that time at a highly excitable or at a not so highly excitable state? And the question is, what can we extract, decode from the preceding EEG that will predict the EMG potentials? And the way that Johanna approached this is by defining a linear measure, which is a combination of a spatial filtering and a temporal filtering. So if you do these matrix multiplications here between the spatial filter, the EEG data in the window, and the temporal filter, you just get a number. And the challenge is to define the optimal spatial and temporal filters that will maximize the prediction. And then if you look at the spatial and temporal filters, maybe you can tell something about what matters to the brain at that point of time. So we have 2 classes, large and or high and low excitability. We use logistic regression to optimize the 2 filters, and then it's very, very nice. What comes out in this subject, it sort of confirms what we've done in a hypothesis-driven way, where we looked at the oscillations of sensory sensory motor mu rhythm. But here it's a completely hypothesis-free approach, where we're asking, okay, what is the most predictive temporal pattern? What is the most predictive spatial pattern? And you can see here that the 10 Hertz oscillation comes out, and the left sensory motor region comes out. So I think this is beautiful work that Johanna has been doing here, and I think this is really helping us to identify the right filters to extract these very basic brain states. So that's project 1. Project 2 is then asking, well, where is the actual anatomical location that matters to these excitability states? And this is another postdoc in the lab, Paolo Bellardinelli, who's been working very intensely in this. And in order to extract anatomical location from the EEG and MRI, it's important to have a very well aligned, high quality anatomical MRI. So a lot of the work has gone into doing these boundary element models, identifying dipoles in the cortical mesh, and then comparing the influence of signals derived from the sensory gyrus and from the motor gyrus. So the precentral or the postcentral gyrus, they're very close together, but it is pretty important to try and find out which one of these is important for determining excitability state. And this is ongoing work, but it looks like, somewhat surprisingly, that the sensory gyrus is more predictive than the motor gyrus. And this is sort of by chance also the region that the electrode C3 that is typically used when we don't have this MRI data available, that preferentially picks out, you can see it here in this average sensitivity profile, that it picks out post-central, so sensory gyrus rather than motor gyrus. So another, I think, pretty important basic question, where exactly is the region that matters? And then moving on to pathways, and now it starts to get a little bit complicated. We're still at very, very basic questions of interacting with brain states, connectivity states. But here, I will try to explain briefly the idea behind this experiment. We're interacting with the pathway from the left to the right motor cortex, and the way to read out the state of that motor cortex is the short interval intrahemispheric inhibition, where the left motor cortex is stimulated with the conditioning stimulus with one coil, and then on the right motor cortex a test stimulus is applied, and the conditioning stimulus, which is 10 milliseconds before, it modulates the response that you get. You can see it in the panel here. It modulates the response of the brain to the test stimulus. And the question we're asking is, okay, so how does the connectivity state or the network state in this pathway between left and right motor cortex influence this measure of short interval intrahemispheric inhibition? We have 5 conditions where the left and the right motor cortex at the time of stimulation are at the positive peak, where they're at positive and negative peak, or the opposite way, or both at the negative peak, or finally the random phase condition. And this is the communication by coherence hypothesis that says that, okay, if the two hemispheres are oscillating in synchrony, then that's the state of the brain where this pathway is more active. So that's sort of the underlying hypothesis. So let's see what that looks like in real world. Here are the 2 coils, and you can already see how a multi-locus TMS would enable us to use bigger coils and go closer together. I think this is really pushing the limits of what's possible with independent coils. These are small, they heat up quickly, but it's definitely one of the more challenging experiments. Here's the EG cap underneath these coils, and this is then the data. Mariana Stefano has been the scientist who worked on this experiment. This has also been published already, so you can look at the details in more depth. Here we're just showing on the left that yes, brain states do oscillate between these in and out of phase states quite quickly, sort of within a few seconds. You oscillate through all these 4 conditions. So the real-time system is able to just wait for a given predetermined condition to occur and then apply the stimulus. And now looking at the results of SIHI, and a lower number here means that the effect of the conditioning stimulus was stronger, so more inhibition of the test pulse. And it turns out that in the in-sync conditions here, positive, positive, or negative, negative, the amount of inhibition is stronger, which is just a nice indication that the coherence state between the 2 motor cortices is an important measure that we can interact with here in a real-time state-triggered way. Okay, so this is a little bit more complicated experiment, and now we're moving to the clinical side again, back to just modifying a single area with a single coil. This is work from Brigitte Renner, who's a psychiatrist, but now in neurology, and she's taken a result that Ulf Ziemann mentioned at his introduction, where the repetitive stimulation during a given phase can increase or decrease excitability, and in the negative peak, this is the condition where we see the LTP-like effects in the motor cortex. And what Brigitte did in this study, and this is a study with patients suffering from depression, that approach was adapted to stimulating DLPFC, dorsolateral prefrontal cortex, on the left side, which is a target used in therapeutic TMS protocols to treat depression. And she compared the condition of an open loop protocol where high frequency pulses were applied at a random phase with the same kind of triple TMS pulses applied only during the negative peak of that area of the brain with a replay condition. This was a single session proof of concept study, so not therapeutic, but more with a neurophysiological readout with 17 patients suffering from severe major depression. And it turns out, this is just one of the findings of this study, but maybe the major one, that after the closed loop condition, indeed, there was a reduction in the power of the alpha oscillation corresponding to an increase in excitability of the targeted network. And here is a very nice demonstration of the difference between pre-stimulus EEG in the closed loop condition versus the random condition of phase accuracy. So these studies are now being similar clinical studies are being prepared for. They're already running for stroke patients as well, and we are extremely excited to use the MTMS technology for some very specific applications in clinical work. And this is the neuro team from the group, and yeah, thank you very much.
Thank you, Christoph. Any questions or comments?
So I can see a question: is EEG-TMS closed loop for higher frequency bands as effective as for the alpha band? So interesting question. We are pursuing work in the theta oscillation in the dorsal medial prefrontal cortex, which is of course a lower frequency band. But the beta oscillation in the motor cortex is another oscillation that interests us, but it's difficult to distinguish the beta oscillation from the first harmonic of the alpha oscillation. So we've had some challenges targeting beta, and theta, however, has been quite promising. And Pedro Gordon is one of the postdocs in the lab who's working on this oscillation. And so thank you to Nivesita for that question. And from Sam Zitman, can you discuss the time lag in the real-time closed loop? So the time lag is not really due to the real-time processing and transport, but it's due to the filters in the system. There's like a 1 millisecond transport delay due to the UDP protocol accepting the network packets. But the Nyquist filters that downsample the EEG at the amplifier stage, that introduces, depending on the filter bank used, a delay of maybe 2 or 2.5 milliseconds if you're doing a low pass cutoff of 1 kilohertz. So those any filters, and filters are important also to extract signal properties. So any filters in the system will induce a time lag, or if you don't use FIR filters, will cause frequency shifts that depend on or phase shifts depend on the frequency. So this tends to be the main thing we worry about. So thank you for that question. And Carola Oram asking as well about artifacts. That is a lot going on with the EEG recording and delivery of magnetic pulse. To clarify, are you able to screen out the artifacts? Yes, again, important point. And we need to distinguish the artifacts before the TMS pulse and the artifacts after the TMS pulse. The artifacts before the TMS pulse are not so problematic. They're the standard EEG artifacts, and those are the ones we need to worry about for the EEG triggered TMS work. And then the artifacts after the pulse, those are those are serious artifacts. And if you want to really extract evoked EEG, you need to do some very specific cleaning. The nice thing is that modern amplifiers, after 50 milliseconds or 100 milliseconds, they're back to baseline, and you can just collect the rhythm again. And I think the last two questions, and in the interest of time, I'll try to be quick. Joris, is there a role of the variation in amplitude of the ongoing algorithm? Absolutely, really important point. There's been some work on this also by Till Bergman. The thing to keep in mind is that high amplitude means better phase accuracy, and so phase and amplitude are always also going to be confounded. We need high amplitude to get high phase targeting, and the amplitude itself seems to also have a sort of complex relationship to excitability. So all very excellent questions. And the last question from Lily: are there any plans for language interventions with stroke patients with aphasia in C2B project? Aphasia is actually one of the symptoms of stroke where we've had some pretty nice success in the TMS clinic here in Tübingen with standard protocols, and we're looking very intensely at language networks as well as neglect on the right hemisphere, which seems to have some similarity. But this is not one of the primary goals, but it's something that is ongoing. So thank you for that question, and yeah, thank you all. It's 1 minute late, so I will shut up now.
Okay, thank you, everyone.
Thank you. Bye.
Bye-bye. Thank you, everybody.
Thank you. Goodbye.
Bye.
Yeah, bye.